She was right. Hello? Hello?
Good morning, sir.
Can you people see my slide?
Yes, sir, we can see it. Morning, sir.
Very good. And you can hear me?
Yes, sir.
Okay, sorry for starting late. So, today we are going to continue in our class. Basically, what we are doing is that we are actually trying to summarize the course itself. We cannot be able to do all the things that we needed to do, or at least we can cover and then be able to do and fulfill all the requirements that are expected of us. So today we'll look at classical genetics and then we'll look at the multifactorial traits. So here, what you are seeing on this slide are classical example of conditions that will actually arise from monogenic disorders. And then, like I said, subsequently, we'll also look at the ones that will arise from multifactorial and polygenic disorders. So what you are seeing here is actually a child that has disorder of the skin that we call ichthyosis. And then this other girl here is a girl suffering from a dominant condition known as the osteogenesis imperfecta. And then a classical scenario is what we call the upper and lower motor neuron disease, which are classical example of monogenic disorders too. So we'll see as we proceed from there. Remember from our history, we actually told ourselves the fact that Gregor Mendel has been classified or has been identified or recognized as the father of genetics because of the series of experiments he has actually conducted that led him to promulgate three laws that we saw in our last class. Okay. And these three laws are the laws of segregation, the law of independent assortment, and the law of purity of gamete. We saw the place where he conducted the experiment and the species of plant, which is Physon sativum, which is called the green beef. And we also saw the characteristics of this particular plant. Sometimes they could actually have round or wrinkle seed. They could actually have yellow or green seed. They could actually have red or white flower, and then they could be tall or they could be short. Now, these are two types of trait, and in genetics, we classify them as dihybrid. So, in the popular Punnett square, crossing of Mendel, what you get to see is the fact that the yellow color is dominant actually over the green color, and the round color, the round seed is dominant over the wrinkle seed. So if you look at this particular crossing, you will appreciate what is actually happening. So if yellow is represented by capital Y, and then green is represented by small y, wrinkle represented by small r, whereas smooth represented by capital R. At the end of the day, when you do the crossing here, you arise or you actually get what we call yellow and the smooth seed, where you have heterozygote Y and heterozygote R. Now, if you take this particular and you break them into the respective genotypes, what you are going to get is to actually live with the two types of the trait, Y, capital R, small y, capital R, capital Y, small r, and then small r, small y, small r, the same thing you do on this other side, and then you try to cross them. So simply carry this Y, you put here, carry this other Y, you put here, carry this capital R, you put here, carry this capital R, you put here. So you do the same thing for all the squares or the cells. By the time you come to the last one here, you will see that everything here is actually homozygous recessive, small letters. So small letter Y, small letter Y, small letter R, and then another small letter R. So at the end of the day, you actually have a phenotype. The genotype represented as 9:3 to 3 and to 1. So this actually goes back to the idea that we were talking about the purity of gamete. No matter how long it takes for a particular trait or phenotype to disappear from physical observation, as much as the genotype comes together, that particular phenotype is actually going to emerge. Okay. So like we said, three laws of Mendel were enacted. Law of gene segregation, which stipulates that when sex cells are formed, the two copies of the gene that controls a trait will segregate or separate from each other so the gamete receives only one copy of the gene. Remember that for eukaryotic organisms, largely they are diploid. And because they are diploid, their sex cells are actually haploid. So the process that will lead to gametogenesis, the production of the sperm or the production of the egg, will actually lead to reducing the number of the chromosomes, whereby we actually have an haploid. So at the point of fertilization, haploid plus haploid will now give us a diploid. Then, the law of independent assortment stipulates the fact that members of one pair of genes do not influence the way in which other pairs of genes are distributed among the gametes produced from the single act of meiosis. Everybody will assort independently. If there is a color, for instance, in this instance, where we have yellow color, green color, wrinkle seed, smooth seed. So when yellow color is actually assorting, it doesn't influence the the other trait, e.g., whether wrinkle or whether smooth. No, they only go to meet by chance, in the immediately formed zygote. And then the law of the purity of gamete, genes are unaffected by the association in the individual, and each genes emerge pure and distinct in the gamete generation after generation. Well, I just took some time to explain that. So technically speaking, depending on whether you are dealing with one particular trait, or whether you are dealing with two traits, or whether you are dealing with three traits, you are going to be having different kinds of ratios. Now, they are simply called Mendelian ratios. So now, if you take your time to look at this particular slide, you'll appreciate the fact that there are quite a number of things that will actually affect those ratios we are talking about. And one of some of those things are listed here. Number one, there may be new mutations that actually arise. Number two, there can be lethal genes. Number three, the degree of the ability of the gene to express itself when present. And then the degree that which it could actually express itself, known as expressivity. So these four things can actually affect Mendel ratio. So when we take lethal genes, for instance, there are certain genes that are actually lethal in the sense that they can lead to the death of the organism. So classical instances are actually listed here. When we look at dominant conditions, we can see hypercholesterolemia, HISS vulgaris, which has to do with the disorder of the skin, multiple telangiectasis, and then in the case of recessive cystic fibrosis, Gaucher's disease, progeria, Tay-Sachs disease, these are diseases or conditions that actually if you inherit these conditions, you may not likely reach a particular birthday. Progeria, for instance, even though there are people who have survived progeria for more than 20 years, for largely people in their teens, some in less than 10 years, will actually die because progeria is a situation where you have rapid aging, so that a child of 3 years could look as if he's a man of 90 years. So, and then you also have X-linked conditions like fibroid disease, Hunter's, Menkes, and Wiskott-Aldrich syndrome. These are classical conditions that can lead to mortality. Classical example are here, ichthyosis, the scaling of the skin. Now, what actually happens in this instance is that the skin, if you remember the anatomy of the skin, the econium stratum now becomes so thickened, and that is what you are actually seeing here. And depending on the type of HRC, some of them can be so thick that any small pressure on them can actually lead to breakage. Then there are what are called semi-lethal genes. In semi-lethal genes, it actually takes some time for the person to actually be affected. So you have classical conditions under dominant trait, Innsbruck disease, or what we call megacolon. And then you have Huntington disease, you have osteogenesis imperfecta, like we saw, on the first slide. And then you have certain conditions that are classified as recessive familial polyposis, Fowler's disease, sickle cell anemia, erythema pigmentosum. Now, at least we are very familiar with sickle cell, and we know that sickle cell under certain times of crisis can be fatal. And then we have an X-linked condition, classical disease or disorders. It will include Duchenne muscular dystrophy, fatal granulomatosis, and then leukemia syndrome. Now, if you take Duchenne muscular dystrophy, muscular dystrophy is actually a disease that is characterized with the fact that the individual doesn't get to age 20. By age 12, definitely speaking, the affected person will be wheelchair bound. And because muscular system is what is actually affected, it may go to affect largely or the principal muscle of respiration, which is the diaphragm. And because of that, breathing can become a lot difficult, and before you know, the individual can start suffering from respiratory disorder that may lead to death. Okay, how about the penetrance and expressivity? Like we said, penetrance and expressivity are scenario where number one, the gene is present, but is it able to show itself? That's penetrance. The disorder is present. What degree of severity is it showing? That is what is called expressivity. And under penetrance, you could actually have conditions that can express between 80 to 100% and between 50 to 80%. Now, classical cases are listed for you here: achondroplastic dwarfism, media, Huntington disease, porphyria, retinoblastoma. And then on this other side, you have 50 to 80% things like cleft lips, congenital ptosis, deafness, neurofibromatosis, polydactyly are classical instances. So what does that supposed to mean? What this means is this. The person can be a carrier of that particular disorder. Okay, maybe career may not be the best word. The person can actually be constituted to express the condition because he's carrying the genotype. But because penetrance is low, the disease does not really show. So the person may look as if he is normal, but the truth of the matter is that he's not normal. So that's where the word carrier may be, likely to be able to explain that. Okay. Now, these are classical instances: polydactyly, which extra fingers; Phocomelia, where there may be problem with the limbs, either missing limb or abnormal limbs, like you can see in this particular instance. Okay, if you look at this particular hand, there are 6 fingers, and the fingers all look normal, so you may not be able to do anything here. But for this one, that is the most common form of polydactyly, usually the 6th finger, which is the smallest here, is usually attached by flesh and connective tissue, so it can easily be cut out. And many people who actually have this have been taken to hospital, and the small finger is removed, so you wouldn't even know that the person is actually polydactyly. Now, if we mention the fact that there are dominant, there are recessive, there are X-linked traits, so how do we be able to distinguish between them? So here we are listing means by which we can use to say, okay, this particular disorder is this particular transmission. So let's start with dominant traits. With a dominant trait, what you actually will expect is that, number one, if there is complete penetrance, the trait will appear in every generation. So it will not hide itself. So in instances where you have 80 to 100% penetrance, there is going to be what? There is actually going to be an expression of the trait. So people can see, or anybody can see, the individual can see that it's actually carrying this disorder. Number two, the trait is transmitted by an affected person to half of his children on the average. Now, when we say half of the children on the average, what we simply means is the fact that it's actually a heterozygous situation where you can have a capital B and a small b, or a capital T and a small t. And then by probability crossing, normal crossing, we know that the percentage is 50-50. But remember, these are just crossing probabilities. So it can actually be 100%. Or sometimes the person may even be lucky not to transmit that particular trait. But theoretically speaking, we know that how the children are expected to be affected. Then number three, it doesn't matter whether the person is a man or is a woman. Both men and women are actually having the equal possibility of transmitting this trait to their offspring. So this is a classical, okay, maybe this is, okay, let's use this as a classical pedigree chart to demonstrate this. In this instance, you actually have the grandfather having this particular trait. Grandmother is free from it, so they actually had 3 children, 2 boys and 1 girl. So the first here is actually having the disorder. He married a lady that is normal, and they had 1, 2, 3, 4, 5 children. And out of the 5 children, 3 were unlucky to inherit this trait. And then the sister here, who had who had who has the disease marry somebody who is normal, and out of the two children you have the first child, which is a girl, being infected. So now, what I would like you to appreciate is the fact that you use that same criteria to be able to appreciate, number one, there is complete penetrance, and because there's complete penetrance, every generation appears to be affected. Generation 1 affected 2 persons in generation 2 and 4 persons in generation 3. So what are the classical example of those conditions? We have seen a few of them earlier on, but then we have added more for us to appreciate. Here, you have the condition, then you have the description: achondroplasia, a form of dwarfism; amyotrophic lateral sclerosis, degeneration of nerve cells or neurons. And this is actually part of the thing that we call motor neuron disease. And cleidocranial dysostosis, lack of bone hardening, polar bone defect, and stuff like that. Gynecomastia, breast development in males, hemochromatosis, iron overload, hypercholesteremia, raised levels of cholesterol in the blood. Then we know how this can lead to some disease situation that can become fatal. So amyotrophic lateral sclerosis is actually a classical condition that has been observed somewhere, I think, in the 40s, in the 30s, and in the 40s, in this particular gentleman known as Lou Gehrig. And that is why the name of the disease is also called Lou Gehrig disease. So this guy was actually a baseballer, and he was doing very well as one of the stars in New York Yankees. And then suddenly in one season of the game, they discovered that his performance was deteriorating, and that is how it continued to deteriorate. At some point, they were concerned they have to take him to the hospital, and by the time they began to study his situation, they now discovered that actually it's not just stress, but it's actually a disease situation that needed attention. And by the time they took time to look at it, they were able to say, okay, since he is the first person to be studied and to be noted and to be recorded, they gave the name of the disease Lou until they were able to give it a more scientific name known as amyotrophic lateral sclerosis. Now you can see that one of the persons, one of the legendary scientists that actually suffered this particular situation is the person that was called Stephen Hawking. Stephen Hawking died some few years ago. You know, he suffered this disease, but that did not stop him from achieving very great things that he did as a physicist. So you can see continuing common examples: intestinal polyposis, manic depression, neurofibromatosis, pulmonary arrhythmia pigmentosa, and stuff like that. These are all classical cases. You can take your time to look at them when you get the slides. Then we have what we call autosomal recessive traits. In autosomal recessive traits, what you actually have here is the parents are usually normal. And because they are normal, they have a risk of actually having 25% of their children affected. If both parents are affected, then it's 100%. Because remember, everything is small letters AA, BB. So if you have small letter AA and the father has small letter AA, as much as they are affected, the children will 100% be affected. Now, one of the things that you should also note is the fact that in this particular transmission, the thread may cross or skip generation. In the autosomal dominant, we say that the thread would appear in every generation if the thread is actually having full penetrance. Then parents may be genetically related, and that is why we discourage people who are related to get married. Essentially, cultures actually have that tradition. They found as scenarios where two, especially when they are closely related, are allowed to mate or to marry. Males and females are equally affected. It doesn't really matter. And affected also in general will have normal parents. And because the parents look normal, they may feel that they are okay, unknown to them that they are actually carriers. All right, so classical cases are listed here on this table, starting with adrenal hyperplasia, albinism, cretinism, phenylketonuria, down to retinoblastoma, which is cancer of one or both eyes. So these are things, and then you can see our progeria that we mentioned earlier, small face, dwarfism, heart disease, wrinkled skin, but all these things characterize what we call rapid uh, aging. So the next is X-linked dominant traits. In X-linked dominant trait, what you have is that essentially you're going to be having affected sons, because by the time you look at the people who are actually expressing the disease, you may discover that there are more of sons than daughters. Number two, heterozygote prime female transmit the trait to half of their children of either sex. Then remember, either sex simply means because whether the child is a boy or a girl, they are going to actually receive the X chromosome from their mother. Then the transmission by female follows the same pattern as an autosomal dominant trait. Cannot be distinguished from autosomal dominant inheritance by the progeny of the affected females, but can be by the progeny of the affected males. If, for instance, you don't know what kind of situation it is, then you may need to actually look at the siblings. Do they have children? Are the children affected? So, how is the transmission in the female sibling? How is the transmission in the male sibling? From there, you'll be able to tell particularly if it is X-linked dominant or autosomal dominant. Affected females are common than affected females in this instance. Now, the reason is this. Oftentimes, these conditions are not compatible with life, because they are fatal. So, majority of the time, the boys don't get to survive. So, but you get to see the girls. And the girls, the reason why they are able to survive is because of the compensatory mechanism of the double chromosome, X chromosome that they have. There is a process known as X chromosome inactivation, where in the women, like the girls in this class as we're taking and they are listening, their body is actually made up of mosaic expression of X chromosomes. If you pick a tissue or you make a tissue out of their body, you put it under the microscope and you see the cells. These cells you are seeing, they are actually one cell or the other cell neighboring the other cell and all over the place on the slide. One is expressing paternal X chromosome, one is expressing maternal X chromosome, and issues like that. So, assuming if the X chromosome is actually from the father, that is faulty. So, it now depends on the number or the percentage of cells that are actually carrying the faulty genes. That would determine the severity of this particular condition. So that is why if the individual is lucky to inherit or to have a high percentage of the normal copy of the genes, the condition may be so mild or even almost absent. So you may even think that the person doesn't have the condition, but anyway. Classical example continues, syndromatosis, Gold syndrome, and the characteristics are there for you to. Then in the case of X-linked recessive, remember autosomal recessive, autosomal dominant, then here X-linked dominant, X-linked recessive. So affected person is more often male than female. The trait is passed from affected male through his daughters to half of their sons. So this is very simple to see. Remember the skipping of generation that we talked about. No direct transmission from father to son because father does not transmit X chromosome to sons. Or the trait may be transmitted by carrier female to half of their sons, like we have seen. Classical example of these conditions are listed here. Anophthalmia, congenital absence of the eye, completely the eye failed to develop. Cerebellar ataxia, atrophy of the cerebellum, clumsy, explosive speech and movement. Then congenital ichthyosis, which we saw earlier on, heart, scaly skin, hyperpigmentation, hypogonadism, hemophilia that we are familiar with, lack of active blood clotting, number 8, ichthyosis vulgaris, fish scale disease, dry, heart, scaly skin. And so you can go on and on and on. Total blindness to spinal ataxia, and then different types of muscular dystrophies. These are classical examples of conditions that will actually be genetic. So now, apart from all these classical instances where we saw autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, there is another way that actually this could be transmitted, and that way is what is called the mitochondrial inherited disease. Now, mitochondria itself carries its own genome, and because of that, um, it could also be transmitted. And the disorders will affect biochemical, clinical, genetic, and then the nuclear. So, and because mitochondria is very, very essential to the survival of the cell, you can see how essential it is. It is the center for energy production in the living cell. And you can see how it affects a variety of organs and tissues in the body. So you can see the eye, the heart. Okay, for instance, take the heart. The heart is one of the most active, very, very active organ. Remember that the cardiovascular system is the first organ system to develop during the process of development. And from that time that the heart may start beating, maybe by the fourth, by the fifth week, the heart will continue to beat until the individual is deceased, possibly at 90 years. Now I tell people, I say there is no machine that man has made that can be as efficient as the heart. Now, because of that, the heart muscular system has a of mitochondria in order to be able to provide the demand for the high demand for the metabolic activity the heart is involved. The brain is another thing, the liver, the muscular system, and quite a number of other tissues are actually listed here for us to appreciate. So quite a number of issues are associated with that. In these tables, you can see neurologic manifestation, mitochondrial disorder, and systemic manifestation, mitochondrial disorders. So if you look at it, you can see ophthalmoplegia, stroke in young persons, seizure, myoclonus, optic neuropathy, dementia, ataxia, myelopathy, dystonia, and then basal ganglia calcification are classical examples of neurological manifestation of mitochondrial disorders or diseases that could actually be inherited through this particular transmission. And then on this other side, you can see system manifestation, cardiac conduction defect, cardiomyopathy, hyperthyroidism, hearing loss, like we just saw, hepatopathy, and down, you can see endocrine pancreatic dysfunction, psychiatric disorders, and classical instances. So this particular cartoon also gives us the idea, mitochondrial myopathy by organ system. You can see them classical, you can see the cardiac heart issues, musculoskeletal, gastrointestinal, ocular, neurologic, and then general. So these are all things that could actually arise from poor mitochondrial performance. And this is essentially because the genes carried by the mitochondrial genome is affected, and because they are affected, they are not able to produce energy, optimal energy that is required for the function of the cell, and because of that, there is going to be deficit, and this deficit will actually result to some of those things that we are mentioning. So how can they, during fertilization, mitochondrial DNA is derived from the oocyte. Or remember, what actually happens is the fact that during the process of fertilization, when the sperm enters inside the secondary oocyte, everything about the sperm is going to degenerate. What will remain is only the head of the sperm that contains the genomic material. So that is why the mitochondria that is around the neck of the spermatozoon is usually degenerated. So the mitochondria we have in our bodies are the mitochondria that we actually inherited from our mothers. So that is how it will be very easy for us to appreciate if a disease is actually mitochondrially inherited. We can trace it to our mother's lineage. That is why we say maternal inherited mitochondrial mutation transmitted only from mother, mutation transmitted to all offspring, whether they are males or they are female, because we receive X chromosome, we receive mitochondria from our mothers. All right, increase mutant mitochondrial DNA in mother blood, increase frequency of affected offspring. Risks of having affected offspring differ between different mitochondrial mutations. So what are the type of mutations that actually have occurred and also the time? So classical example, we have seen those ones before on those in tables and stuff like that. Another set of situations like that. So remember three major classes of transmission. One, autosomal, which are divided into two, recessive and dominant. X-linked also divided into two, recessive and dominant. And then the last category is called mitochondrial disorder. So in mitochondrial disorder, we have seen that we could only inherit that from our mothers, because the mitochondria in our body is actually the mitochondria from our mother. So what is that supposed to mean? What it means is that your mitochondria, they are the same mitochondria like the mitochondria in your maternal uncles. Why? Because your maternal uncle share the same mitochondria with your mother. Because they also inherited the same thing from where? From their mother. So your maternal aunt and uncles, you share the same mitochondrial destiny with them. So if there is a disease that is transmitted via that, then you'll be able to see the disease also affecting your maternal aunts and uncles. Okay, that is just a brief. At the end of the day, when I'm sending you the slide, this is not the slide I'm going to send you. I'm going to send you a more elaborate slide. But for this lecture, we're taking this so that we're able to do some quick work. Now, before we move to the next topic, which is all part of this basic classical genetics, is there anybody who have any question to ask? Question? Okay. If there's no any question, we move multifactorial or polygenic traits, just like we say. When we talk about multifactorial, we are talking about the fact that genes can actually interact with the environment and produce a particular kind of phenotype, or instances where we need more than one gene to produce a particular effect. So when we talk about that, or some other thing that we may also need to appreciate is the fact that multifactorial traits do recur in families because they are partly caused by genes and shared environmental factors. So if there is somebody in the family who has a particular trait, don't be surprised that trait is also going to appear in another member of the family. And that is what we mean by a recur or recurrence. Now, this is the spectrum. You have genes on this side, and then you have environment on this side. In between the genes and the environment, then you can have different types of expression. And here you have genetic diseases, major gene multifactorial, complex multifactorial, and then environmental, which are essentially infectious diseases and injuries. But then you can see in between these, you can see major gene multifactorial, A, BRCA1, BRCA2, actually breast cancer. I mean, genes that predisposes to breast cancer, familial hypercholesterolemia associated with cardiovascular disease. Remember that cholesterol, while the body metabolizes, produce and stuff like that, but we essentially consume it from the environment. That is why we tell people to stop eating this stuff or reduce eating this, reduce eating that so that you don't get predisposed. And then the other one here again, you can see complex multifactorial caused by genes and environment. Alzheimer's disease, asthma, diabetes, most cancers, and then that's why we tell people to stop smoking, to stop drinking, to stop doing certain things, to increase activity and all kinds of things. You know why? Because this can actually uh, be taken from the environment in the sense that your body and the environment can interact. And when we say interaction here, we mean through your lifestyle of eating, lifestyle of drinking, lifestyle of living, any kind of life, interacting with your genetic makeup, leading to the production of this kind of disease conditions. So how will we know that, okay, the condition we are seeing is actually multifactorial? Number one, a single organ is usually involved in the expression of that trait. Number two, gene expression appears to be triggered by an environmental agent. If the environmental agent is there, then it will be triggered. Three, sex is relevant to the risk of the demonstrating the trait. So you may discover that the trait may be influenced by the sex of the individual, oftentimes, maybe more in the females, maybe more in the males. Then the risk is higher when more than one family member is affected. So if you have more than one family member affected, then there's also likelihood that other family members will be affected in the future. Then recurrence rates depend on the severity of the expression in the affected trait. What is that supposed to mean? What this means is that, take for instance, cleft lip and or cleft palate. If you look at somebody who has a cleft lip or has a cleft palate, how is the severity of the cleft lip or the cleft palate? If it is just small, uh, the likelihood that it may actually express again in another member is low. But if it is severe, it gives you the idea that this thing may actually recur in the family. And that is why we say that the recurrence rates depend on the severity of the expression. Consanguinity of parents moderately increase the risk of their offspring expressing or coming down with this multifactorial. What is that supposed to mean? We have cousins getting married. Naturally, we are increasing the risk of their offspring coming down with that. So we discourage people actually from doing that. Twin studies have shown that the trait has a genetic component. So two twins, if they are really exposed to the same environment, they are going to be both. If you remove one of the twin from that place to another place, because the environmental trigger is not available in that environment, the person may not be able to demonstrate or express that particular condition. Then number 8, the risk decreases sharply with each outward degree of relationship. So as you move away from people related to you to go and marry somebody from far, so you are actually reducing and reducing and reducing the risk. So while in the actual sense, in our respective cultures, especially in Africa, we encourage people to marry from. In fact, I was just listening to one podcast that one man did from my place, and he was worried about the fact that the young men will go and bring their intended fiancé spouses to their families, and they will tell them that no, this person is not from this place, is not our tribe, and issues like that. You say, okay. Then they will say, okay, the person will go and bring another person that is close by. They will say, still no, we are not really satisfied. Then they say, okay, the person will go and bring from the tribe. And then they will say, no, this person is coming from that part of the clan, or is coming from that other one, and stuff like that. So the question he asks is, okay, so what do you want the person to do? The person should go and marry angels or what? So this is actually a cultural thing that you find not only in Nigeria, across the world. People are comfortable with them marrying from within their own people. But genetically speaking, we are supposed to go outward. Okay. Moving forward, threshold model of this kind of scenarios. In multifactorial are quantitative will continue distribution. And how can they control such disease as scabies, spinal bifida, and issues like that? So that is how what we are going to actually see. What actually happens is that there is a threshold for the disease to actually manifest. And how will the disease manifest? See how it happens. So usually in a bell curve shape, as you move from the right, from the left to the right, you see the liability actually increases, and there are some points which will cross the threshold, and before we know it, the trigger will kick, and then the person can actually express. So you can see here the number of affected people towards the right. Now, we need to appreciate the degree of relationship so that we can, because remember we said that the occurrence of this trend actually will decrease with movement. So where you have first degree relatives, you are actually having children, their parents, they form the 50 because they share 50% of their genes in common. Number two, second degree relatives share 75% of their genes in common. This will include our aunts, our uncles, our nieces, our nephews, and then our grandparents. Then third degree relatives will share 12.5%, and this will include our first cousins. Okay, so most affected children have normal parents. In fact, because of the normal parent, that is why we feel that we are okay. Everybody will say, I'm good. Everybody is good. And that's why you can see very handsome man, very beautiful woman. By the time they get married, then they will have a child. A child that may be affected with any kind of threat, including multifactorial threats. So that's why we say oftentimes, they are actually having normal parents. Most geniuses come from normal parents. Most mental challenges come from normal parents. So it's the same kind of scenario. Recurrence risk increases with the number of affected persons, children in the family. We have already stated that several, but not an unlimited number, loci are involved in the expression of the threat. There is no dominance or exclusivity at each of these loci. The loci act in concert in an additive fashion, each adding a distracting or detracting a small amount from the phenotype. So what is that supposed to mean? The more the number, additive in the sense that, like we said, remember the title of this particular segment of the class is multifactorial stroke polygenic. Polygenic meaning many genes. So the more the affected genes are being added, the more the severity of the condition. And then the environment interacts with the genotype to produce the final phenotype. So these are the things we must keep in our mind. Now, they could actually be classified as quasi-continuous multifactorial phenotypes. And if you look at this table, you'll see the group. Remember, one of the major issues is that an organ is actually affected. So congenital heart defects. What is the organ here? The organ is the heart. Arterial septal defects, coarctation of the aorta, patent ductus arteriosus. These are all anomalies of the development of the heart, which we did in embryology. So hole in the heart, narrowing of the aorta, failure of the ductus arteriosus to close, so persistent ductus arteriosus. Then coronary heart diseases, hypertriglyceridemia will actually result atherosclerosis, essential hypertension. These are all things that will lead to increased blood pressure and stuff like that. So this is a classical scenario to show some of these things. Number one, pulmonary stenosis, thickened narrow pulmonary outflow. Number two, thickened right wall ventricular wall. Number 3, ventricular septal defect. Number 4, overriding of the aorta, and then septal defect. These are all the things that we can find in classical condition that we call teratology of Fallot. All right. The other thing we need to appreciate is the fact that the head, the feet, the mouth, the eyes, and the ears. For instance, classical case can be stuttering. See, when you hear people stuttering, you may think that it's just it's a very complex situation. That is why oftentimes you see the person who as he's talking, then you see hitting his head, hitting his feet on the ground and hitting the table, for him to talk. It's a classical instance of what? The classical instance of, multifactorial speech delay in articulating letters and words. Joints, congenital hip, rheumatoid arthritis is a classical case of dislocation of the hip, adverse swelling and stiffening of the joints. Mental state like Alzheimer, so you see Alzheimer disease can be transmitted in different ways, including multifactorial. You get the logic. Manic depression, manic depressive psychosis, which is actually severe alteration of moods. It's a classical case. So, brain in subject with Alzheimer disease. What actually happens? So you can see all the issues. 1, immune system and inflammation, DNA damage and repair, metabolism, oxidative stress, and neurotoxicity, intercellular communication within the nervous system impaired, apoptosis and cell death leading to Alzheimer, cytoskeleton and membrane protein disorder, endothelial function and cerebrovascular insufficiency. All these things can actually lead to what? Can actually lead to Alzheimer. And don't forget, these are things that could actually be caused by environment and interacting with the genes and stuff like that. But the resultant effect is what you are seeing as brain in subject with Alzheimer's disease. And what actually happens is the neurodegeneration of the, or is the degeneration of the neurons, simply known as neurodegeneration. So in the case of the person who is normal, you can see active neurons firing, whereas in the person who is suffering from Alzheimer, you can see the neurons are dying out and then they are degenerating. Then there is accumulation of neurofibrillary materials. All right. So some of the other issues are also listed here. You can see mental states, schizophrenia is a classical case of multifactorial threat. The spine in instances of anencephaly, meningomyelin, scoliosis. Now, spinal bifida that we saw from the beginning of this particular segment, is a classical case of this. So, stomach, colon, and kidney, you can see classical case pylori stenosis, which is narrowing of the pylorus. They are all classical instances. So you can see here, the spinal bifida with meningomyelin completely the bag is open. The spinal cord has not fully developed. Now you can see the other one, spinal bifida occulta, where you can see that, okay, there is a problem here, but then you cannot see the spinal cord. And then these 2 other pictures down, and they are actually anencephaly, where the brain actually failed to develop. Neural tube close the cranial part of the body, they fail to actually close, and because it failed to close, the brain failed to develop. So these are classical instances. Anomalies of the nervous system when there are various distinct. You can see anencephaly, encephalocele, encephaly for the spinal cord, spinal bifida, dysplasia, meningomyelin, and meningomyelin. So classical instances that you can actually see. So when we talk about recurrent risk, what do we mean? Recurrent rates is defined as the probability that a defect or disease will reappear in the relative of an affected person. Now, what will cause that? The relationship, the number of affected persons, the severity of the expression for the threat, and then lastly, the sex of the affected. Remember, we said that the sex of the person actually have a role to play. So that even if the threat is going to appear next, likely thing that is going to appear in a particular sex, if it is in a boy, or in a girl. Okay. So let's move to some other concept. Some other classical concept will include things like sex-linked characteristics, sex-influenced gene or autosomal, then sex-limited. So sex-linked characteristics, genes located on the sex chromosome, which we have seen, and then sex-influenced genes and genes on autosomal chromosome that are more readily expressed in one sex, and then sex-limited autosomal genes whose expression is limited to one sex. So what is the difference? The difference between influenced and limited is the fact that both sexes can actually be affected, but the degree of expression will be influenced by whether it is in the males or whether it is in the females. Whereas this one can only be seen in one particular sex. So sex influence autosomal trait that is expressed more often in one sex than in the other. Now, on this particular instance, you will appreciate what we are saying here. More common in males, cleft lip, and palate. You can see the ratio. Before you get to see one girl or woman that has cleft lip or palate, you have seen two men. Congenital pylori stenosis, you will have seen 4 men before you see one woman. Gout autosomal, 8 men before you see one woman. Pylori stenosis, 5 men before you see one woman. More common in females, anemia. You will see actually 4 women before you see one man having anemia. Anencephaly, you will see 3 women before you actually see one man. Okay, actually, let me say that you don't even see any man or woman because anencephaly is not compatible with life. But when you get to see, a stillborn. Now, before now, you know, people are taken unawares. But now with the technology that we have, where mothers will go to antenatal and can go and do organic obstetrics, ultrasound, and stuff like that. If it is detected that a child is already having an anencephaly, there's not much to do. So the best thing to do in the Western world, what they will just do is to actually terminate the pregnancy and bring out the child. You know, instead of carrying the child to term that you know is not actually going to be alive. So why don't you just do a surgery to remove the child? Then so that the mother can recover quickly, and then at some point, if she's thin or if the family think they can still go ahead to have another consumption, subsequently that will be better for them. Lupus erythematosus. Before you see one man, you will have seen 9 women. And then manic depressive psychosis, osteoporosis. We are very familiar with osteoporosis, and that is why in our physiology we have been told that, see, women, when they reach menopause, one of the risks they actually get to face is the risk of they are vulnerable to fractures. And what leads to fractures? The fractures result from the fact that the bones have become weak and brittle. So any small pressure can actually lead to breakage or fracture. So we are not surprised to actually see osteoporosis. But then let's go back to number 2, anencephaly. Why is it more common in female fetuses than in male? Now, you see, the idea is that anencephaly is when the story gets too bad to this extent. And what we get to see is that there is a continuous abortion of the male fetus. And because of that, you don't even get to see at the end of the day, or you just get to see a few, just like it's reflected here. Now, sex limited is sex limited. Remember, we say it can only appear in one sex. Classical example: formation of breasts, ovaries in female, testes in male, prostate in males. Partial here is actually expected to be seen only in males, even though under certain disorders, like in the case of hirsutism, you get to see women developing here in androgenic sensitive areas. Production of eggs in the female, sperm in the male, hypopadias. These are classical instances that are very common in a particular, or they are seen in a particular sex than the other. Now, the other thing we need to talk about is what we call pleiotrophy. Pleiotrophy is the influence of a single gene on more than one trait. So what does that mean? It has multiple expression. So if you look at this particular instance, you have gene A. Gene A, because it's defective, can produce trait 1, trait 2, trait 3. Classical instance here is actually the sickle cell gene. You have the sickle cell gene here that is affected, then affected, then pleiotropic, phenotype that will lead leading to blindness, liver failure, heart attack, and stuff like that. So these are the multiple traits. So any single gene that can generate a sequelae of traits, simply known as trophy. Multiple alleles. We are very familiar with that because this is a situation whereby a single gene occupying a given site or locus, on the chromosome can have alternative states or alleles, because over time in the DNA occur that actually in the DNA occur at that particular locus. So this is actually nature's way of increasing the whole thing. So here you actually have classical cases like the blood, classical, which all of us are familiar with because we have done blood physiology. We know that blood actually are different types of blood. The classical one, the ABO groups. Beyond that, there are some other blood groups that can characterize individuals, but these are the major ones, and all of us we know. And all of us can classify ourselves, under AB or AB. So, and what actually makes the difference is the presence of, on that particular locus of the blood. What is it? And that's what you can see here. If you notice how the blood is AB, AB, or O. And what we actually have here is that you can see AA or A. And then this one, there's nothing there. Then here you can see BB or B, or for both is A, one is A, one is B. And then the antigen that will produce, and then the antibodies that will actually react are actually listed here. So classical case of multiple alleles, classical case of the blood group. Phenocopy. Phenocopy is another interesting thing that we must also know, because here we are talking about a scenario where a defect can be caused by environmental factors, but then they look as if they are actually genetic. So this will actually lead to malformation, deformation, and disruption during the process of growth or development in the womb. So phenocopy traits are not transmitted to further generation because they are not actually genetically constituted. So that's one of the things that we must treat in our minds. Fine. Then the other thing that we must also tell ourselves is the fact that we need to be able to construct pedigree charts. A pedigree chart is a pictorial representation of family history, essentially a family tree that outlines the inheritance of one or more characteristics. So we say, okay, just like we have seen previously, we saw the distance. So the symbol commonly used in pedigree chart are summarized in the substance right here. All of you should take your time to be able to appreciate this. Where we have a square simply means a male. A circle means a female. So if we write a number inside, it gives you the number of, if we don't have space, we can put a square and then write two daughters. Put a square, boys, We put a circle and write 3, meaning 3 girls. Now, when we put a square or a circle and we put a small arrow by the side, we mean problem. Problem means the first person that will notice the problem. And when we cross the square or the circle, we mean the person is dead. When we shade it, we mean that the person is affected. And then you can go on and on and on. Then some of the other things that you can actually do is you can see unknown sex, lived 1 day, that after birth, the person died. Then stillbirth, we don't know the sex, miscarriage, or if the person is actually pregnant, we can indicate by this. And then several other ones they are here. So take your time to be able to memorize, to appreciate, so that if you are going to draw a critical chart, you know exactly what you are doing. So here you can see a career of excellent traits. Usually when you draw the cycle, you put a dot inside. Then take your time to look at it. So when we say what is a critical chart, shows a record of family, one individual. They can be used to study transmission of hereditary condition. They are particularly useful when there are large families and good family record over several generations. Very important. We need to appreciate the fact that records are very important for you to be able to achieve that. Okay. So a classical case is actually shown here. Remember, we have seen number one, all the generations must be aligned to label as the one generation, one, second, third, fourth, depending. You have your family history, you can actually let the people out. Then if there is a condition that is transmitted, you identify the people who are showing or demonstrating, you draw them up. So in this instance, we can appreciate what we are seeing. See that in first generation, the grandmother or the great-grandmother is affected. In the second generation, both the children, both the daughter and the son are affected. In the third generation, 4 people affected. In the fifth generation, 4 people affected. So simply, when you see something like that, you can clearly declare that it's actually an autosomal dominant trait, as you can see at the legend. So what are some of the things that we need to consider when we're actually looking at this? We need to consider things like the ethnic history of the individual, familial versus non-pattern Mandela inheritance, determination of family related. Remember, we just the second and third relatives, degree relatives, then consanguinity are people related, then chronological age and what age, then the most likely reason for a genetic disorder can also looked at. So now let's look at this other one. Here we have generation 1, 2, 3, 4. First generation, nobody is affected. Second generation, 1 person is affected, the daughter here. Third generation, nobody is affected. Fourth generation, 1 person, the child, and son is affected. So this is a classical instance where we say because there is a skipping of generation, we can clearly say it's autosomal recessive trait. So genetic diseases associated with Negroids. So remember we said we need to look at the ethnic background of the individuals. So blacks in general will have higher risks for lactosemia, lactose intolerance, and then sympathy. So their definition is their inability to convert the lactose to glucose, tendency to bleed, susceptibility to infection, and then lactose intolerant, inability to digest lactose, and then sympathic fusion of two or more fingers, and then their mode of inheritance, whether as autosomal dominant and stuff like that. And then continuing hemoglobinopathies, hypertension, polydactyly, tuberculosis, uterine fibroids. These are classical cases of conditions that are more frequent or prevalent in the blacks as compared to the whites. In fact, I remember reading a paper many years ago where some people were proposing a theory. They say one of the reasons why the African Americans actually in the United States have higher prevalence of blood pressure than the white people there, they were trying to relate it to the kind of situation and pressure, physiological stress they passed through when they were being transported from Africa to the Americas. In the Hebrews, the report have actually indicated that congenital pylori stenosis, Gaucher's disease, Tay-Sachs disease is more prevalent. So you can see poor development of death follows if the narrowing is not corrected. Enlargement of liver, lymph node swelling of the body, and liver damage in Gaucher's disease. Tay-Sachs disease, degeneration of vision and motor skills due to accumulation of lipids, and death follows about 2 to 4 years. The other one still in the Hebrews is what we call phenylketonuria, hepatitis media, cystic fibrosis, porphyria variegata. These are classical cases of conditions associated with then congenital or disease and genetic condition due to consanguinity, that's mating closely related individuals. Albinism is a classical case. Febrile ataxia, phenylketonuria, Tay-Sachs disease, erythema pigmentosum. These are classical instances where you actually have. And then the percentage contributed by this, you can see as much as 40%, contributed as a result of consanguinity. You see, lipid disorder, death before the age of 5, very common in the Ashkenazi Jews, also the so the age of onset, remember we talked about chronological age. So now, one of the things you must also keep in mind is the fact that you cannot just declare that the disease does not actually run in a family if the people are not able to read the age at which the disease can actually manifest. So club leaf, palliate, and club food, you see bleaching will be seen at conception and at birth. 1 year, from conception to 1 year, you can see it. Cystic fibrosis will also show. Histiocyte congenital will show. Osteogenesis imperfecta will show. Familia will show. Sickle cell will show. Then, the other one that you also need to appreciate is the fact that by 10 years, you will need to get to by 10 years, Gucci muscular dystrophy, myasthenia, Hunter's disease, osteogenesis, tuberous sclerosis. By 20 years, Alzheimer and start presenile psychosis. Then diabetes insipidus, diabetes mellitus, pernicious anemia. By year 40, amyloidosis, diabetes mellitus, familial polycystic kidney gout, which is adult pneumonia, then Huntington disease, then so forth and so forth. So by 50, by 60, other disease actually emerge. So when you take osteogenesis imperfecta, this is what you get to see. There are different types of osteogenesis imperfecta. So this actually would describe when we talk about severity or expressivity of the condition. See, this is type 1. Female age 38 years, height 171 centimeter. Type 4, male 40 years, height 90 centimeter. Type 3, female age 27, height 94 centimeter. Type 3, male age 40, height. So you can see that the bones, that's why it's called osteogenesis imperfecta. There is a poor formation of the bone. So the bone will become mangled as a result of the defective calcium and the rest. So you can see that the first person on this side has a milder expression of the disease compared to this person on the other side. So this is a classical condition. I mean, this is a classical way of seeing the issue of expressivity that we mentioned. So these are pictures of people who have mild and then who have severe. Completely this one, they cannot even move. Have to be actually sitting on the chair. So osteogenesis is a spectrum of disorder from mild and then to severe. Okay. So low frequency variance and susceptibility, genome-wide association study are proven adept at identifying common variants contributing to inherited components of the disease. Almost all such variants seem to have modest effect size, and even when combined, their impact on an overall population variance and predicting power is limited. But today, we actually have a technique that we call genome-wide association studies. In this kind of situation, you can actually study a whole population at once, so that you are able to actually identify all the genes and the possible reasons why able to actually cause the effects and stuff like that. So we have actually come to the end of today's class. So in the next class, we will find from there. Looking at formulating, then looking at the screen and something like that. So remember, I told you earlier on that we are just doing a general revision, of genetics. So you take your own time to look for books, for instance, if you can get books which are all over the place or on the internet. You can actually source material to complement questions. You ask questions so that we can actually answer them. Issues that you need clarification on. Anyway, so are there questions before we call it a day?
Oh, good afternoon, Prof. Hello, good afternoon.
Yeah, good afternoon.
I don't know, you mentioned a class before this one. I don't know, is it last one we had, there was another class on genetics.
What? You say I mentioned what?
That there was I don't know if there was another class on history of genetics that was done before this one.
No. You see, this class that we are taking today, there are actually two topics. That's what I meant.
Okay.
Yeah, one is classical genetics introducing Mendelian inheritance, then the other one is multi and polygenic traits.
Oh, okay.
Yes. That's what I meant. Other question. So who is Ashampa and Denira in this class? Huh?
Okay, so I think he has just been added to the group recently.
Okay, Ashampa.
He's one of the MSc students.
Okay, I can see he's raising his hand. So because I'm not sure
Yes,
I've
sir.
met him before. I've not met him before.
Yes. Okay, resumed quite late.
Ashampa, you have question? I can see you are raising your hand before.
I just wanted to identify myself.
Oh,
I'm the latest addition to the group, sir.
okay. So what are you studying?
MSc,
MSc. Okay. Okay. So where did you do your own degree, first degree?
University of Maiduguri.
Huh?
University of Maiduguri.
University of Maiduguri. Ashampa. So wannan wani irin suna ne Denira. Where are you from?
I'm from Taraba.
I see. Okay, you are. No, but I met you. I thought. Okay, are you from University, Federal University of Ugari?
No.
Oh, okay. You are coming on your own. Okay. So well done. Well and welcome. All right. If there is no question, we'll call the class to hold, and then next week we will I'll be a little busy, but I will squeeze time to see that we take at least one class next week, because I have exam on Tuesday, I have exam on Thursday. So I will see if maybe on Wednesday or on Friday we can take a class. Okay. Thank you.
Okay, Prof. Thank you.
Have a good day.
Yeah, thank you, sir.